I would like to welcome those who have just joined. We'll be starting today's webinar in more 10 minutes. Thank you for joining. And meanwhile, we begin with today's webinar. It would be great to hear from you about your educational background and also your research interests. So you can put them in the chat box. Looking forward to it. Thank you. Thank you everyone for joining. We'll be just starting in 2 more minutes. We are waiting for others to join as well. Thank you. I would like to welcome you all for today's webinar, where we'll discuss about our upcoming summer programs and the opportunity to get trained and work on an independent research project with mentor guidance. I'm Sonalika Ray and I'm the Omics Logic Community Manager, and I'm responsible for daily interaction with thousands of our users that are a part of our bioinformatics community that is managed by Pine Biotech. To talk about the flow of today's webinar, we'll first discuss about Omics Logic Metagenomics Training Program, which will commence on May 23rd. Metagenomics is relatively a new field of research based on high throughput sequencing or next generation sequencing, and its popularity signifies a growing appreciation for the composition of microbial communities and the genetic information their genomes carry. Metagenomics allows us to study the microbiome in its natural environment and learn about the cooperative and often symbiotic relationships between prokaryotes, eukaryotes, viruses, and archaea. In this metagenomics, omics logic specialization, we will explore how different types of analysis can be used to understand the microbial world. We will cover several bioinformatics approaches to the analysis of the microbiome. We will learn to compare microbial composition in different conditions, learn about the sequencing of viral samples, and discuss how microbes evade the immune response of the host, as well as evade treatment. We will do so in the context of several projects taken from high-impact publications. Next in the webinar, we'll discuss about bioinformatics for infectious diseases, which will commence on May 17th. This program is dedicated to studying the viral diversity and its role in epidemic infectious diseases that keeps re-emerging, including zoonotic spillover, transmission between hosts and humans, and the process of viral and bacterial disease development. The participants will get a chance to learn about bioinformatics and analyze genomic data by applying various analysis approaches to study viral genomes. As a result, you will learn to understand the relationships between viral strains and haplotypes, find differences in sequence data, and see the implications for drug and vaccine design. This program will provide opportunities to practice analyzing data to gain hands-on experience with curated datasets from public domain collections, which are present in these example projects, guided by experts with bioinformatics experience and knowledge about virology. Later, we'll also discuss about other upcoming summer programs, which includes precision oncology, Omics Logic Genomics, Cheminformatics for biomedical drug discovery, and also we'll discuss about the summer research internship or the fellowship program. We would discuss about these programs schedule, how joining these programs in summer can help you get into your dream degree in the fall or upscale your skills for better career opportunities, and finally, how you can join these programs and learn from experts. The Omics Logic training has been completed by over 24,000 participants from 187 countries in over 300 workshops, And due to this fast growth, our team is working with local and regional coordinators that are helping refine the local program logistics and leverage our online training resources, adapting them to the needs of students and researchers around the world. So now let's quickly sign up on the Omics Logic Learn portal just to make sure that we have the access to the resources that we are going to cover today. So I'll just share the link to the portal once again and share my screen. Yes. So here is the link in the chat box. Please make sure that you all have an account on the Omics Logic Learn portal so that it's easier for you to follow the resources that we are going to cover today. I'll just quickly give you a demo on how you can have your account on the Omics Logic Learn portal for free. For those who already have an account, can just put one in the chat box, and others can follow the process along with me just to make sure that you have an account. And if you face any technical issues, then we are here to assist you. So you can just simply click on the login button on the top right, and then you'll get some options by which you can have your own account. Either create your account by putting in the credentials that are required, or simply sign up with any of your social media accounts. I'll be using my Google account for the same. Please let me know in the chat if you have an account already, and if not, then you can create one and let us know that you have done so. All right. Next, what we want to do is to have an updated profile. So the next steps for those who have already created their accounts or are doing so, please update your profile. You need to click on your mail ID in the menu bar itself. This will lead you to a profile section. Now, joining these programs will give you an access to the Omics Logic resources, which includes the coursework and also the session recordings. So it's very important for you to go through these resources and, most importantly, monitor your progress. And also having an updated profile helps us monitor your progress timely. So please make sure that you have an updated profile picture. Your name should be in title case. This is the same format that will reflect on your certificates that you'll get in the profile section itself once your particular coursework is complete. Please make sure that you link your social media accounts and also add a brief bio about yourself that could include about your educational or research background and also your research interest, so that it's easier for us to guide you and also for the mentor to know about your research interests. It's very important to make sure that you have the starter subscription active as a part of the program, and also the badge of the program should be added to your profile. So that was what I wanted to discuss with you about signing up on the portal and also updating your profile. Now you are all set to go through the resources on the portal. I would now like to invite Mr. Ilya Brodsky, who is going to take you through these programs, metagenomics and bioinformatics for infectious diseases as well. Ilya is a CEO and co-founder and has a background in bioinformatics, data science, project management, and commercial product development. Mr. Brodsky has facilitated and also managed collaborations with researchers at Stanford, UCSF, Louisiana State University, Tulane, and other academic and pharma R&D teams. He also oversees many of these scientific projects to talk about this important area of metagenomics analysis and sequencing. Now, I would like to pass on the stage to Mr. Ilya. Over to you.
Thank you very much, Anarika. And so what I'll do in my part of the presentation, I'll start by introducing the Omics Logic program, and then I will pass it on to Dr. Raghav, who is going to be speaking about his detailed review of the curriculum and the types of exercises and projects that we will conduct. So the Omics Logic Metagenomics program is a program designed to introduce the participants to the role of the microbes, but also to the practical aspects of data analysis to understand from data generation to processing to downstream biological interpretation. So what's important about metagenomics is that metagenomics is different from many other types of omics because it is trying to characterize a community instead of an individual. So what this program does is it is organized into several different sections. We will first cover the logic why we are trying to ask specific questions, how do we ask specific questions, what are the results that we're expecting to get. Then we will talk about practice. How do you practically try to apply these methods of analysis and prepare for independence? And that leads us into the project where you can identify a data set and plan your analysis and execute it with the help of technical support. The logic focuses primarily on the different types of data that metagenomics covers and the specific questions that we would be asking using that data. Then the practice leads us into the specific analytical approaches so that we understand in detail what are the types of analyses and what are the outcomes of those analyses, and how do we know what is significant, what is not significant, where is signal and where is noise. We have to understand that so in bioinformatics, there's an environment where this kind of an analysis takes place. And that includes being able to design sequential combinations of code, which are called pipelines. And so those pipelines provide us with the outcomes that we can then try to interpret the composition of the microbiome in the environment. How does the environmental or the gut microbiome can be associated with behavior? Can we look at the gut microbiome, the skin microbiome? Can we identify associations with clinical factors, etc.? So leading from the very basic understanding of the logic all the way to the application where you can start analyzing specific data sets and ask these important questions. The program is organized into several sessions that are separated by the type of application, the type of project, or the type of analysis that you're going to be doing. Now, for those of you that might be completely new to the area of metagenomics, I want to encourage all of you to try to complete the free introduction, which is called Bytes and Molecules on the portal. So Sonalika already introduced you to the portal. If you have an account, this is a free course, and you will see there that one of those is an introduction to metagenomics. So in that course, you will be able to understand the terminology and the specific types of applications of metagenomics. What we will do is then later on, if you would like to prepare, is you can try to go through the Metagenomics Data Analysis course. And in that course, we cover data generation, types of projects and data sets, and all the way into the specific analysis. And what we will do in the program is we will actually take a look at the logic behind the analysis, why these methods were created, how do we apply them, what do they actually produce, and we will take a chance to practice all of these analyses. First of all, using our point and click interface called the Bioinfo platform. A good example that I will demonstrate here in just a second is the data to pipeline. So the data to pipeline is divided up into several stages, from pre-processing to then actual connecting, so denoising, what's called denoising. Data to stands for denoising. And then assignment of taxonomy and visualization. And then we will use this pipeline to actually understand different types of visualizations. So let's just briefly take a look. On your portal, on your profile, you will find that with that profile, you should have access to our platform, and this is the link. And this platform combines many of the standard tools that we typically use for processing and analysis of data. And if you follow my example, I pasted the link in the chat, you will see that there's a whole section here dedicated to metagenomics. And that includes 3 different sections: metagenomics or microbiome, metagenomics using KIND2, and Amplicon 16S and 18S, which looks at data to and denoising the reads so that we can assign taxonomy. So in each one of these sections, we have a demo pipeline. And the demo pipeline actually allows us to understand the analysis as we build the pipeline. So going through this interface, we're able to actually see how to connect the individual methods into a pipeline. One second. Why this is not loading? Okay. Let me actually try this again. Okay. So after you complete this pipeline, you will also find that there's visualization that makes it easier to start looking at the data and understanding the composition of each sample, comparing the samples in their proportionate abundance, and understanding the differences between the different types of samples that we have using a variety of statistical and analytical methods. So like here, we can see NMDS. So in this program, and briefly, I will just mention several of the important kind of aspects of the microbiome. We will learn about microbiome in the human body, including the skin and the gut or the stomach, where you will understand the different types of compositions associated with different kinds of phenotypes. We will look at how these bacteria form communities and how we can analyze these communities to understand the relationship between a specific phenotype that we can interpret in a clinical or a phenotypic way and the actual composition of the microbes. And finally, we will look at collections of data sets where you can sample a specific type of samples to ask questions. So in conclusion, this program is going to give you the tools and the logic behind the analysis so that you can be independent in asking research questions and conducting the analysis, getting the results that you can publish or potentially put into a poster presentation. So with that, I will pass on my screen to Dr. Raghav, and he will talk in greater detail about the curriculum of this program. So Narika, please make him a co-host. He's not a co-host, so he can't unmute himself.
Thank you very much. Finally, I am a co-host. Thank you very much, Sonalika. And let me get find ways to share my screen. Okay. Just loading it up for me. Okay, see my screen? No, it's still loading. Yeah. Yes, I think it should work. Can you see my screen?
Yes, sir, we can see.
Yes, thank you very much. Yeah. So thank you, Alea. I am Raghavendran. I am bioinformatics mentor and research consultant with Fine Biotech. And I will take you, I take this opportunity to welcome all of you for this webinar on metagenomics analysis and study on microbiome and also for bioinformatics for infectious diseases. So we understood that metagenomics is a way to study microorganisms in their natural environment, dissecting community structures, exploring species richness, and investigating functional roles of these microbial communities, functional roles that these communities or different microbial communities play in many areas related to human health for human health and diseases. So in this program, actually, I will discuss what you will learn in the upcoming Omics Logic metagenomics program that is going to start by 23rd of May. And this online program will actually cover main aspects of metagenomic sequencing, analysis of 16S rRNA, and other aspects of metagenomics and microbiology research using bioinformatics. So at the end of this program, participants will be conducting independent or group projects analyzing data mainly from public domain and to learn how high impact biomedical publications are possible on the microbiome subjects. So let's go over the syllabus in detail to get an overview on the variety of different bioinformatics analysis, the logic behind the analysis, and different downstream processing methods you will be that you will be introduced to in this specific program. So we start with an introduction to next generation sequencing for metagenomic sequencing, and this could include whole genome sequencing, and this could also include 16S rRNA sequencing and amplicon data types also. So we first start with the overview of tools and resources that is necessary for this program, like you will be given an introduction to various courses, projects, data sets, how to access the associated resources, how to access the Bioinfo server platform, and how to get access to different projects and different example projects that we are going to discuss through this program. And then we follow it up with an introduction to metagenomic sequencing and its application in studying microbial community in functional bacterial community analysis. So here we are going to understand about whole metagenome sequencing and how to understand or how to identify different different phylogenome or phyla or different variety of microbial community from mapping onto databases like Silva and NCBI, etc. So we will also deal with different data formats that is required for performing such analysis, and we will also understand the general flow of metagenomics pipeline, and we will also get a practical hands-on on variant analysis in Bioinfo server and how to visualize generally the mapping and the mapping of the reads and mapping of these reads to the whole genome reference genome. So and then we will go on to visualize 16S metagenomics data. So here we are going to understand the microbiome variability using amplicon sequencing, and we are going to also use 16S rRNA sequencing for the identification of microorganisms to study microbial community. We will also discuss a couple of data from human gut project and also data from acne, or how to understand the data distribution of acne data from NCBI bio project. And what follows is an hands-on project example on human American gut and human microbiome variability. So here we will demonstrate you how the use of small subunit rRNA sequencing is performed and is used for identification of microorganisms, and we will also introduce you to the definition of gut-brain axis and microbiome and its effect on behavior of the hosts and different downstream analysis methods that can be performed after the completion of pipeline analysis on the 16S amplicon data from Bioinfo server. And then what follows will be a detailed project study, an example project study, which I will also demonstrate, show it to you in the Learn Omics Logic platform. So this is about metagenomic study on how the gut microbiome is affected by diet, for example, a high-fat diet and a normal diet, and how this diet has an effect and has an influence on the behavioral aspects of a mouse system in this case. And you will also learn hands-on practice on performing these downstream analysis in, let's say, phyloseq, using phyloseq library in R. You will also have different access to different visualization methods that is used to understand the taxonomic composition, and you will also given separate space to learn and practice phyloseq analysis code in R. Excuse me, it's not responding for a moment. Yes, I got it. And then we will complete the metagenomics courses by metagenomic courses by culminating into giving you information about how to plan a bioinformatics research project. So all that you have learned so far is going to come and is going to end up in making you develop your own project from publicly available data. We will show you, or we will guide you how to explore and how to extract data from such big data repositories, and how to carefully and design analysis methods that is tailored for your data, and then how to perform this analysis as you had been learning throughout the program, and then how to get a good biological interpretation, and how to understand the biology and different phenotypical characteristics of the sample that you have been using for the project. And throughout the duration of the program, and for duration of your project, what follows after the program, you will have access to mentors for discussion on your research progress, and you will have access to mentors and different people for troubleshooting your technical support on a regular basis, and you'll also get suggestions and expert feedback on your progress whenever it is required. So let me help you, or let me give you an example by showcasing one of the projects that we recently prepared on the role of high-fat diet on obesity. So before going into what are the different types of analysis possible possible for such a project, let me give you an overview of what is a gut microbiome and what follows with the analysis of microbial community within the gut microbiome. So in healthy adults, the gut microbiome exists in a state, or in a mutual symbiosis state with this host. So this environment of the gut dictates both the composition and the functional productivity of the adult gut microbiota. And this gut microbiota may interact with the host through presentation of different ligands, such as molecular patterns that are associated with pathogens, and different metabolites that are produced. And these different molecules are responsible, and these different molecules will modulate the immune system or immune homeostasis of the gut gastrointestinal tract. And as you can see in this comparison, the gut microbiome will have an enormous impact on the life and also on the stages of the host. So it plays a fundamental role in actually shaping the composition and function of the gut, and the way the food is processed, and this becomes key factor in determining the amount and the type of material that is reaching the host's circulatory system as through the growth and production of microbiota metabolites. So in this perspective, we can see that the relationship between symbiosis and dysbiosis of a gut microbiome in a healthy individual and an obese individual will differ by their, for example, difference in the variability, or difference in the different types of microbiome that is available, or that one observes in the healthy human gut or healthy gut. So to study this in this specific project, what was actually performed is a mouse system or mouse model system was used to, and it was fed a high-fat diet and a control diet, and their response to a normal diet and their response to a high-fat diet is studied, and also some of the microbiome is actually transferred from the high-fat diet mouse to the normal diet to understand whether or not this high-fat diet microbiome that is transferred into a normal microbiome will influence their behavior changes. So to generally to learn about what sort of analysis that can be performed with this data, let me quickly stop share here and go over to the Learnomics Logic platform. I will find ways to where I can show you I can find this. Okay. Okay, can you see my screen on project 13?
Yes, sir. We can see your screen.
Yes. So after performing background statistical analysis, we can get a lesson on how to perform hands-on, how to set up a pipeline for hands-on analysis of the 16S amplicon sequencing data. So in this lesson, you will be guided in a step-by-step process through how to take the raw data and then how to perform these analysis pipeline and how to set up the pipeline and how to perform the pipeline and what are the different settings that is required to get the pipeline running and what are the different outputs, output files that we are going to consider for downstream analysis. And in the downstream analysis section, these major output files are used to perform downstream analysis using PhyloSeq and using basic R tutorial. So this tutorial on how to perform downstream analysis on this amplicon data will include a detailed learn the code section where each and every part of the code is being explained to you, and then what follows is different data visualization based on which we can understand what the data actually tells. So here the data tells the distribution or proportional abundance of phyla between the Heifer diet and the Chow diet is different for the two different mice. So that is what you will progressively see when performing this analysis using that is displayed or that is represented, visualized using different visualization. So you also will have access to try yourself these analysis methods in a special try it yourself area. Let it load. Yes, where you can learn from this code and then perform these analysis yourself in the try it yourself section. So again, this measure of diversity like alpha diversity, beta diversity, which will also tell you how different these two mice are because of the difference in the proportional abundance of microbiome that characterizes their gut area. So you will get, as I mentioned, a detailed explanation of all possible analysis methods that you can perform, and you'll get a try it yourself section where you can try these analysis methods with specific question that is directed to you after learning the codes. So with that, I will actually pass the stage to Elia one more time.
Thank you, Raghav.
Unmute myself or mute myself. Yeah, thank you.
Okay, so the second topic that we wanted to discuss today was bioinformatics for infectious diseases. Before we go on, if you have any questions throughout the presentation, please feel free to post them in the chat. After the end of this presentation, we'll be able to address any of your questions. So, like Sonika mentioned in the very beginning, in this webinar, we're going to be talking about metagenomics, but also program specifically designed to train those who are interested to apply bioinformatics and study infectious diseases. So briefly in the next few minutes, we'll talk about this program. I will pass it on to Dr. Raghavendran to discuss the specific curriculum and example projects that you will be able to work on. And then after that, we will finish the webinar by talking about the summer research programs that we have planned. So bioinformatics for infectious diseases is really a program designed to address the pressing need to understand, characterize, monitor, and address by intervention the infectious diseases that are so prevalent and are characterizing this 21st century. So in this program, we'll talk about how to apply data analysis tools, and a lot of them are going to be somewhat similar to each other, but applied in very different types of outbreaks, epidemics, coronavirus, Ebola virus, rhinovirus, tuberculosis, malaria. So different types of viral, bacterial, and other pathogenic diseases. The program is really designed in a similar fashion. You will explore relevant topics, tools, methods, data repositories, project examples, and you'll be able to see how multi-omics analysis of genomic, transcriptomic, proteomic data is going to inform different types of decisions about outbreaks. Before we jump into this, let's briefly talk about what is the definition of infectious diseases. So infectious diseases, also called communicable diseases, are called by a pathogen, and that pathogen can be a bacteria, virus, parasite, or fungi. And these kinds of diseases are different from non-infectious diseases, or what's commonly known as chronic diseases. And so we will actually talk about specific applications of bioinformatics to the study of infectious diseases. Now, in the last couple of years, all of us have been focused on COVID-19, and I think really as a result, we kind of see infectious diseases primarily caused by viruses. But in reality, they could be caused by different types of pathogens, including bacteria that can colonize and kind of subdue different types of functions that are important, but also can release toxins and cause organ failure. So in the recent years, there have been a number of pathogens like the Ebola virus in 2014, the outbreak that brought to the forefront genomic surveillance of epidemic outbreaks. There have been diseases that have been affecting millions and millions of people. And we see that these diseases actually are not just the ones that are new, like novel coronavirus, but there have been for centuries, like malaria. And malaria affects a huge amount of people and has been for many, many decades or even centuries. Novel diseases caused by the coronavirus or by re-emergence of specific diseases are going to continue and be very important and dangerous situations that we'll have to address using such tools. Now, importantly, these kinds of outbreaks are only one type. They are kind of silent, and then all of a sudden there's an outbreak. But there are many viruses that cause or lead to chronic diseases. And so even though we kind of tend to separate these two categories, there is a quite large overlap. For example, Epstein-Barr virus and other types of viruses can lead to cancer. So in addition to that, right, beyond viruses, there is a whole field studying different types of bacteria. And for example, drug resistance, microbial or resistant to antibiotics is a huge concern. And so in this program, we're going to talk about examples in all of these different fields. And importantly, we will also talk about the response to these diseases that our body already possesses and how different types of treatments like vaccines are leveraged to actually enhance this kind of a response. So when we look at the history of infectious diseases and we look at the methods to control such outbreaks, you will be surprised to find that actually vaccines that we talked so much about today are not the only ones that were responsible for the reduced load of infectious diseases on the population. So from a population health perspective, we can see that the establishment of health departments, municipal use of chlorine in water, use of penicillin are all predecessors of the vaccines, which was introduced in the 1950s. And up until today, those methods were actually very, very effective. However, what happened just recently is all of a sudden, after having such success with methods that we already discussed, COVID-19 has surpassed the mortality of even such diseases like cancer. And so on one hand, we have this dynamic where we have effective measures that are quite simple. Just washing your hands, having chlorine in water, having availability of antibiotics. And then all of a sudden, we see this tremendous growth of mortality caused by a virus that has already been studied. And so that leads us to the significance and the impact of bioinformatics. So in the era of COVID-19, not only were we able to analyze that data quite rapidly, but very quickly we were able to identify a specific pathogen that then was tracked almost in real time to understand how it changes, how it evolves, how it starts infecting more and more people, and starting to ask important questions. And the ability to ask those questions using data that was collected is the essence of bioinformatics. What is bioinformatics? Bioinformatics allows us to leverage these data sets that have been collected, ask specific questions, and get to a practical interpretation so that we understand something and we're able to make conclusions. Now, importantly, this pandemic has opened a number of new questions that are now being asked, and we will see how those questions are being asked by researchers that have the expertise to design experiments or to leverage data that was collected from others. So here's an example where we can see that we can not only detect a specific strain of a virus, but we can actually track it over time, and important questions will start to emerge. What is the long-term consequences of COVID-19? How do we monitor and prevent outbreaks of these infections? How do we leverage our detailed genomic characterization of the virus or the pathogen and introduce a very specific targeted therapy that can prevent or kind of make it less dangerous? So with this brief introduction, I will again pass on the stage to Dr. Raghav to speak about the specifics and examples that are relevant for this upcoming program, and answer any questions that you might have about what are the outcomes of such training. So over to you, Dr. Raghav.
Thank you very much, Elia. Let me again share my screen. So hello once again. So in this session, we are going to discuss about how different or various big data bioinformatics analysis methods will actually help you in understand the bioinformatics of infectious diseases. And we will also, in addition to discussing the application of different bioinformatics analysis methods, this program will also show you several examples of these analysis methods that are used in pathogen diagnostics, in evolutionary studies of the virus, and in the discovery of antiviral drugs and vaccines, and the design of antiviral vaccines, as well as to characterize emerging drug resistance across the different pathogens. So let's go over this briefly, go over the syllabus briefly. So by the end of the program, you will be able to explore relevant tools for characterizing sequence variation and sequence alignment at the whole genome level. You will be able to learn from peer-reviewed publications and example projects about how phylogenetic analysis helped in understanding the emergence and spread of pandemic like COVID and epidemic like Ebola viruses. So you will also be able to master key analysis methods and visualization tools that are related to sequence variation with functional properties of the protein. And these tools will help you understand the mutations that are critical for the pathogenicity and are critical for the functioning of the pathogens. You will also get an introduction to study the host's response by analyzing the transcriptomics data of the infected host compared to the healthy host. So we can also understand how actively the host is responding to the pathogen or to the infection in this case. So we will start again with an introduction to next-gen sequencing, and we will discuss about various NGS sequencing methods, and we will discuss about different data formats that are used in the sequencing, whole genome sequencing data, and we will discuss about exploring the Bioinfo server and exploring visualization of the reads that are mapped onto the whole genome sequence, in this case viral sequence or pathogen sequence, using visualization tools such as JBrowser and etc. Then we will follow this up with getting an introduction to multiple sequence alignment methods and phylogenetic tree analysis to understand the viral adaptations and the mutations that are characterizing the different pathogens. So we will also work with an example projects and examples of deadly mutations in Ebola and how that translates into different virulence or different effects of pathogenic effects of Ebola virus. So we will also get to compare multiple sequences at amino acid level, at nucleotide level, and also compare different methods for constructing phylogenetic tree analysis. We'll visualize the phylogenetic tree results using specific software that is available for tree analysis and also for understanding multiple sequence alignment methods. So in this hands-on session, this will be a combination. This will be a follow-up hands-on session to the session that you studied before and discussed before. So where you will be shown how to build a pipeline so that you can perform multiple sequence alignment of viral genomes and a phylogenetic tree analysis using different phylogenetic tree methods. So you will also be given an introduction to different analysis pipelines, like selecting appropriate genomic sequences and selecting different pipeline methods that is suited for either FASTQ files or suited for FASTA files. So we will also analyze the output, discuss the pipeline results, and analyze how to perform the downstream analysis of the pipeline outputs and what to do if we have a FASTA file and FASTQ file for input. And we will also understand which databases to use to gather information regarding the viral sequences, and we will also see how to interpret the phylogenetic tree analysis that is the result of different phylogenetic tree methods, and how to understand the evolutionary relationships between the genomes in terms of evolutionary time, in terms of mutation, and in terms of birth and death of different strains that are with a varying degree of pathogenicity. And then from there, we will go to understand how viral transmission occurs cross species, and we will take this, or we will understand with the help of Ebola virus and what are the specific insights we learned from studying the epidemic of Ebola, which is, as Elia mentioned, as one of the first epidemic that is studied or that is real-time tracked using next generation sequencing data. And we will also understand the origins of these viral adaptations, and we will also extend this analysis to novel coronavirus genome sequence so that we can compare the novel coronavirus SARS-2 viral sequence with the other related viral sequences. And we will get a good introduction to how the disease progresses once the virus infects the host. So what happens and what are the functional domains of different proteins that, for example, spike proteins and different proteins that are involved in the disease progression and involved in helping the virus getting access into the cellular different cellular cytosol, and how this virus uses or hijacks the host cellular mechanisms to enable its own progression. And we will also understand how this how the disease how the pathogen evades the immune response of the host system in this session. And then what follows is a hands-on session of an example project, and this example project will compare the SARS-2 whole genome sequence with the other related viral sequence of SARS, MERS, and other to understand how related SARS-2 novel coronavirus whole genome sequence is with the other viral sequences. We will also take SARS-2 sequence from different hosts so that we can understand the zoonotic origin of SARS-2 and how SARS-2 is related to, for example, to different parts of SARS-2, how it is related to different hosts like from bat, from pangolin, etc. And then we will also understand how to analyze the mutation types, and how to analyze different mutation types like synonymous mutation, missense mutation, non-synonymous mutation, and how to calculate the rate of this mutation, and how to map these mutation rates onto the spike protein, for example, or onto the protein, so that we can get a visualization or hands-on visualization or exploring the protein structures, and that is color coded, or that is mapped on for the mutation rate at its different position. So we will use use of Chimera and softwares like Chimera, Chimera X, and PyMol for this specific visualization. And then we will also understand what are the different low frequency mutation, and how will they actually enable the virus to maintain its virulence, and what are the different annotations on what are the different locations on the genome and proteome, different protein that contains these mutations, and how it relates to the accumulation of mutation only on the specific areas of, for example, spike protein and other related proteins, so that the virus adapts itself to maximize its virulence and remain fit in the evolutionary pressure. So finally, we will also understand the host side or host response to such pathogenic infection. So we will get an introduction to transcriptomics data analysis. We will get an introduction to what is measured from the host side, and how we can perform a comparative RNA-seq pipeline analysis, so that we can identify key responses from the host side for the pathogenic interaction. So we can understand how different, for example, healthy hosts and infected hosts differ in terms of their immune response, and what are the different factors for considering vaccine design when after identifying a specific host's response pathways. So this program will also culminate in helping you to design a bioinformatics research and bioinformatics research project. You will be able to choose your topic for your project, and then again you will be explained where to extract data for example, related to infectious diseases and related to specific viral repositories. And then we will also get a lot of hands-on training on how to perform this analysis over the course of program that you can apply onto your own project. So I want to highlight 3 related publications that is published in 2022, and with the available number of data that is deposited in the public repositories for COVID. And these program and these analysis methods will enable you to learn, or will enable you to understand how these studies are performed, and then you can generate your own research question in the very relevant pandemic era, and which still helps you to understand different aspects of SARS-CoV-2 infection, and how big data bioinformatics is generally helping us tracking this pandemic, and also preparing us for any future attacks in the upcoming years. So we all know how the SARS-CoV-2 data, the rich data, has enabled understanding the different variants of concern or variants of interest, what are the different relationship between these variants, and how in real time, how these phylogenetics is being tracked, and how these different variants are related to each other. And we also can get an introduction to how the virus actually enables itself, or how this virus actually has an intra-host variation level that is not being explored in detail previously. So to get a scale of, to get an understanding of on the scale of genomes that is being tracked with the COVID-19 response. So within a month of infection, we got almost an average of 1,300 genomes were being submitted to NCBI and being submitted to GISAID database per day regularly. And within the 6 months of start of the pandemic, GISAID had almost like 110,000 full-length SARS-CoV-2 genome sequences. So by the end of December, by the end of 2021, 2 years into the pandemic, we had 67,000 genomes that were deposited to the public viral genome repositories per day, daily. So currently, the repositories like GISAID, GenBank, NCBI, Virus, and COG UK, everything, all of these combined to contain over 6 million SARS-CoV-2 genomes in its entirety. So we can see where the phylogenetic studies and different surveillance studies are performed all over the world, and irrespective of different continents, all the continents contribute to tracking this viral pandemic in real time, and that is possible mainly through the analysis of big data analysis of the next generation sequencing data, and that has enabled us to understand and also keep exploring the data for future patterns and for further patterns that is so rich in its variety and throwing us a lot of surprises. So one of that is one of that surprise, or one of that key discovery is the in-host diversity of this virus, or its ability to adapt itself to in-host within the hosts based on the host response. So what this means? This means that the viral sequences that for example, that causes severe symptoms, or whose virulence has an increased rate of virulence, has a different variation compared to the virus whose virulence has mild symptoms. So understanding this in-host diversity will pave way for us to arrest this development or this increase in the virulence factor of this virus, and thereby, even if the virus spreads from the host to the interhost or different to the different individual, we can actually make sure that not the most virulent virus is going to spread and going to cause infection for the other diseases. So we also get an overview in what is being currently studied and what is being clinically valued in the current pandemic scenario. For example, we value a lot on genomics and transcriptomics data only about the SARS-CoV-2. And from the virus's point of view, mostly the genomics data is what is being considered really informative. But we have a whole plethora of omics data from the host and also from the viral, from the pathogens responses, which can be integrated into a big multi-omic studies. And that aspect of this infectious disease scenario or pandemic scenario has not been explored in detail so far. So there are still a lot of opportunities there to develop methods that will effectively integrate, for example, the host's response to the pathogens and also the virulence levels of the pathogens to the host response, to study how these two partner in the effective transmission of the virus. So we have not even touched about how or what are the long-term effects of, for example, COVID infection. So into the third year of the pandemic, and we have still a lot of data that can be studied, for example, for the symptoms that prevail, what happens after 1 year or 2 years of 2 years of the infection. Already there are studies that that try to characterize the fatigue level, that try to characterize the loss of smell and taste level of the host individual after several weeks and several months of COVID infection. So this pandemic and also this infection has actually thrown up a lot of opportunities which are being currently explored at a different degree and an increased rate. And this program will enable you to first understand and dissect all of these analysis methods and then develop an analysis method for your own for different data sets so that you can understand the biology of these infectious diseases, or you can understand, apply these methods to another disease of your interest and and gain a lot of experience in the bioinformatics analysis of infectious diseases. So with that, I will attend some of the questions if you have any, and pass the stage back to Sonalika or Ilya.
Yes, Sonalika, if you are there, you could maybe describe some of the other summer programs.
Yes, Ilya. Thank you so much, Dr. Raghav. And I'll now quickly share my screen first of all and take you all through the registration process for the two programs that we have discussed. And then we can go through the summer programs as well. And if you have any questions, please put them in the chat so that we can answer them. Or we also stay back after the meeting is over and take your queries. Yes, so talking about the infectious diseases program, here is the link to the program page in the chat box. Now, how do you register yourself and where do you find these resources that we discussed today about the session schedule as well? So visit the program page, and if you scroll down, you can see there is a calendar where you can see the dates for the scheduled sessions and also the details about each and every session. So you can see the title and the topics that would be covered. And along with each and every session, you are supposed to go through some of the resources. Now, these resources are actually the lessons on the Omics Logic Learn portal to which you'll get an access to as a part of the program. And along with each session, you are supposed to go through the session recording of the session that would be uploaded on the program page once the session is complete. And also you need to go through the associated resources. And on the date of the session, you can see a complete session icon that you need to click on to complete a particular session. And you need to follow the same practice for each and every session that is mentioned on the program page. So that was about the schedule and what we're going to cover for each and every session, and also the associated online resources. I'll shortly take you through these lessons and show you how they look like. We have a question in the chat box, so I'll just pause for a second. Yes, you can definitely register for the summer program after 27th May. So these programs are going to run asynchronously. If in case you're not available before 27th and you want to do it later on, you can definitely enroll. The programs would be active till July 31st, and you can go through the asynchronous session recordings and also the resources on the portal. And we'll connect you with the mentors via Slack channel where you can clarify your questions anytime. Kartika, for the metagenomics course, the timings of the sessions the live Q&A sessions would be around 10 a.m. CST, that would be 8:30 p.m. IST. And in case you're not available for the live Q&A sessions, we can take your questions in a separate meeting as per your availability. And you can go through the session recordings, as I said, at your own pace, as the recordings would be available on the program page itself. For the Slack channel, we are going to share the URL, the link, basically, the link of each and every Slack channel with you. Once you're registered, you will get the link to the Slack channel. You can then join the Slack channel where the mentors are also a part of these groups and clarify questions. Yes, definitely, Basundhara. We do work on research projects that you can work on and get published in reputed journals. We can discuss about the research fellowship program as well, but just shortly once I'm done with the registration for infectious diseases program, metagenomics, and discuss the summer programs as well. All right, so coming back to infectious diseases registration page. Now scroll down and you will be able to find three different subscription levels. Now what are these? So you have three levels here: beginner, intermediate, and advanced. Now in the beginners level, you'll get the access to such live sessions and also the resources. Definitely the resources, access to all of the resources remains the same. It just varies on the type of mentor guidance that you will be getting throughout the program. In the intermediate session, you will be getting the access to one-on-one live sessions with the mentor as well. And taking the advanced subscription will help you work on your research proposal. Like you can go through the program and then have a live session, a live meet with your mentor and discuss about your research question and have a proposal in hand that you can further work on in your research, like as your research project, and definitely have an outcome. Either get it published or present your work in a poster presentation competition in the conferences or symposium. As Ilya mentioned, we are going to have a symposium in the month of August as well. So you can definitely present your work in an international symposium like that. And many of you have criteria to also submit your project if you're a student, to submit your dissertation work in your college. So yes, that thing would be justified with the advanced level of subscription. So that was about infectious diseases program. Now, one thing that I would like to mention here, you can simply click on the select option button and then proceed with the payment gateway. In case you are in a country where you're not able to proceed further with the select option icon, you can reach out to us, and we have Sparsh here present as well in the meeting, and you can reach out to her at marketing@omicslogic.com. Sparsh, can you please put your details in the chat box? And she'll be able to assist you with the alternate options that you can choose to get yourself enrolled. So you will have a smooth enrollment process. We'll ensure that. And now let's talk about the metagenomics program. So the link is in the chat here. Thank you, Sparsh, for sharing your email address. And also Sparsh has shared the contact number that is active in India. So if you are in India, you can call her and clarify any of the questions that you might have. All right, so talking about metagenomic data analysis program. Similarly, you can also look at the schedule, the session schedule of the program on the program page of Metagenomics Data Analysis. And for each and every session, we have mentioned the topics that would be covered and also the associated resources, the lessons that you're supposed to go through. These lessons consist of the course material, which includes the content. It also includes the guidelines to run various pipelines on the Bioinfo server that you should try to practice yourself, and also get your outputs and understand how to interpret these outputs to extract some meaningful biological insights. And similarly, we have all the session details for each and every session present here on the program page. And to enroll for this particular program, you need to pick any of these subscription levels. The criteria remains the same for beginner, intermediate, and advanced level. You pick the level that works the best for you. And yeah, that's pretty much how you can enroll yourself for the program. Now, I would like to pass it on to Sparsh so that she can clarify your questions, and then we will look at more of the summer programs that we would be having in the month of June. So Sparsh, over to you.
So hello everyone, I'm Sparsh and I'm working as Business Development Associate at Pine Biotech. Regarding the registration steps, as Sonalika said that if anyone who is not able to proceed with the payment checkout with this select option, so we can specifically send them an invoice directly on their mail ID, or we have specific UPI IDs for Indian contacts. so as to make the payments more feasible. Also, we have bank transfer details for both outside India as well as for Indian interested contacts. So those who will be facing any difficulty while enrolling themselves for the program for metagenomic data analysis or for the bioinformatics for infectious diseases can contact me on marketing@omicslogic.com, or I have provided with you the contact information. It is active on WhatsApp also. So you can connect with me via WhatsApp, as well as Indian and outside the Indian contacts also. So I'll be there to help you and assist you out. If still you have any questions related, you can simply write down in the chat box and I'll be there to assist you with. Thank you.
Thank you so much, Sparsh. And do we have any questions for Sparsh? If you are interested to enroll in the program, and if you have any questions, you can please ask her. Yes, Anag, you can definitely switch over to the research fellowship program to continue and publish your work once you have your research question in hand. All right, so if we have no more questions okay, we have a question. These all programs are scheduled and it is there on the Omics Logic Learn portal, Avinash. So if I'll just show you, when you visit the Omics Logic Learn portal, there's a tab called Programs over here. So you need to just click on the Programs tab and you will find a list of these programs, ongoing, upcoming, and you can explore them. And if you click on any of these programs, then you'll be able to learn more about it. All right, so with this, now I would like to tell you also about the programs which you can join asynchronously anytime. So one of these is Transcriptomic Data Analysis program, and the link to the page is here. I'll put in the chat box. Sparsh, can you please put your number once again? Vasundhara wants it. Can you put in the chat box? Thank you. All right, and similar to the pages that I showed you before, this page also works the same way, where you can look at the session details for each and every session and then pick up the subscription level that you would like to pick and enroll for it. And the next program that I would like to discuss about is the Genomic Data Analysis program that you can also enroll in any time along the summer and leverage from it. Now, on the page itself, you will find the session details once again, and also to enroll, you can scroll down and pick the subscription level that works the best for you. Now, as many of you discussed and asked also about the research fellowship program. So what is this program all about? And like, what is the structure of the program? For how many months can you enroll? What outcome can you expect by enrolling the particular research fellowship program? You can find all these details here on the program page. So if you are a student and you are looking forward to do your internship for a 2 months time period, which actually starts from June to July, maybe, or July to August, as per your universities or institutions guidelines, then you can join the 2 months research fellowship program. But it is more feasible to join the 3 months or the 6 months research fellowship program, so that you can get better outcomes. So in the 2 months of the research fellowship program, it would be possible for you to do a proper literature review and then also work on your research project with some preliminary data analysis. But a 3 months research fellowship program would be most structured, where you will also get to finalize your research as a poster and present, as I told before as well. You can present your poster in any of the conferences and symposiums. And if you are dedicated to get a publication out of your research project, then definitely 6 months time period would be the best for you, where you can validate your results, work on your manuscript with the mentor, and finally look for a good journal where you can publish it and go through the process that is required. And to learn more about the mentors of the research fellowship program, you can visit the page here. And also on the page itself, you will be able to look at some of the research fellows presentations and also to look at the project submissions by some of our research fellows. I'll show you the exact place where you can go and also look at these project presentations that came out very beautifully by our research fellows, and they were able to publish their project on our portal. So if you go to the Omics Logic Learn portal and click on the projects tab, you will be redirected to all these projects which were put together by our research fellows. These research fellows were from Amity University campuses, India, and also a lot of them were from various universities in in US, Africa, China, and you can go through their project publications and learn more about how this program works and what type of outcome is it possible for you to have as well. So very briefly, I have tried to mention about the whole research fellowship program, but if you're interested to learn more about the whole structure, how it exactly works, what is your research interest, if you want to tell us, and then if you want to understand how the whole training process and the research part will work for you in this program, then please feel free to reach out to us. We can have a one-on-one meet maybe where we can make you understand the whole process and align it as per your research interest. Giselle, the link for genomic data analysis is here in the chat. All right, so do we have any more questions?